thank you for the welcome to country. That's a great presentation for us to think about the land that we're on. Now, I have a question. You might have forgotten since Oliver spoke first. Has anyone got soil on their place? Yeah? Yeah. Have you got a spade? Anyone got a spade? Do you keep a spade in the car, the truck? Do you use it? Except for digging yourself out of a bog or something. So do you look at your soil? Do you dig a hole? You don't have to go all the way down, even the little surface, just to have a look. But, you know, the other thing is that I come from Western Australia, and we've already heard that Western Australia's got sand. Well, actually it's soil, and one thing I've learnt being over there is the subtlety of sand, you know, and farmers have a lot of variation on their farms and their properties, and they understand that. So I just say here, you know more about your place than I do. So what I would like you to do while I'm talking is think about your soil. And like it's very different here in this environment, and some of what I'm going to be talking about is a more productive system. But you know lots about your soil. You know what you can do with it. I don't. But I'm going to give you a talk about soil biology, and that's what I've been playing with for a long time. And again, it's about plants. So we've heard that from Colin. So just in summary, the animals, these are from a farmer that we did a workshop, but so I'm going to talk about plant-soil interactions and soil health. And what is that actually, what's in that? Soil biological fertility. That's a phrase that I started using. Plant diversity, inputs, management, and then throughout it, it's about measuring, because a lot of people ask me, how do you measure what's in soil? I want an indicator. I want to know what's going on. I want to know if my soil's good. And I can't give you an answer, and you'll hopefully understand why that's the case. And then the Soil Health app, and this is a little logo that later, when you've got Wi-Fi, not now, but you can download the Soil Health app. Has anyone seen the Soil Health app? No? Okay, so I'll come to that at the end. So the research, I've been at UWA for a long time, and a lot of people have contributed to our research on soil biology. I started out mainly working on mycorrhizas and with pastures. It was funded by the Australian Wool Corporation. They don't call themselves that now, but they supported my research on soil biology in 1974. And we have kept that focus on pasture management, organisms, what's going on in soil. It lends itself to cropping too, but the wool industry supported me in my career, so I really am grateful, and it's great to be on this property and say that here. Now, soil diversity. I want to highlight that, because especially in the context of someone wants to know, is my soil good? Is this a good soil? Well, it depends, because soils are very diverse. Now, here's some sandy soil from the Swan Coastal Plain in Western Australia, not the agricultural broadacre area. And here's some clay soil, and to get a good picture of clay soil, I went to northern New South Wales, and good picture of clay, worms and all that. But the highlight here is that soils are different. So what you expect, the processes in all the soils are similar, but what you expect to measure in these different soils will not be the same. So we've got soil diversity, that affects what the biology looks like. Plant diversity, and Colin has explained that extremely well. And then you've got the microbes, and then you've got animals, all these things that are living in soil, but they depend on these things. But they are key, we've already heard that. So these soil, plant and microbes and animals, they all interact. So I'm just sort of saying that in order to pick the sweet spot, the actual measurement of how this whole thing is working, it's very difficult, and a spade is pretty handy. So you get interactions, and if you look at what you've got in soil, we've heard already, a lot of mineral particles, sand, we've got a little bit of organic material, that's the roots and the organisms in soil. It's only a small amount, but it is very important. And it's very important that we manage the soils so they can do their work, because that bit is what's key to having the healthy soil. Oh, wait a minute. I can. So Colin talked about diversity, and I agree that having plant diversity drives soil diversity. Whoops. What happened there? I suppose I could stay on this one. Maybe I'll just use this. Can leave it. Okay. So I've been working at the University of WA Farm, which is an hour and a half out of Perth, and I've been introducing perennial grasses, not native grasses, but perennial grasses into annual pastures. And commonly in Western Australia, in the cropping area and the grazing area, apart from the coast, annual pastures. So we want to keep cover, we want to keep as much plant material on the soil as possible. And a lot of our annual pastures can disintegrate and leave very little very little cover, and especially if they're overgrazed. Well, we don't want to overgraze, but sometimes that does happen. So multiple species builds the soil biology, but what do we actually want? So when we're growing canola or wheat endlessly, we really have a decline in that diversity of organisms. So there is a diversity. I'll go over here. There is a diversity of organisms in soil, but we can't measure all these things. It's what's happening collectively that actually contributes to soil health. So the animals are doing their thing, they're coming and going, the other organisms are coming and going. We've got our little pores in soil. This is a sand, a lot of pores in sand, and that is good and bad, because our sandy soils can lose their water, they can lose their organic matter, because the organic material in a sand is very exposed to microbial degradation. It's not protected well. So roots, mycorrhizal fungi, part of this whole system, the hyphae spread into the soil. But you've got all these things going on, and you actually, in the science world, you have an expert on all these different bits. So you've got an expert on one, another, but what you are, you people, are an expert on the whole lot, because you're managing at it all as a whole. And we can only know about the bits. You can integrate and bring it all together by using plants, by managing grazing, by dealing with the things Colin was talking about. And what that is actually managing is this. So you are amazing managers of very complex systems. Now, when I started research in soil, I thought of this word fertility. Now, when you look at the literature, and when I was a younger person a long time ago, you look up the literature and fertility is all about chemistry. And I'm teaching soil biology at the University of Western Australia in this very good soil environment with lots of physicists and chemists and biologists, especially rhizobium biologists. And I'm sitting there thinking, how does biology fit into fertility? How? Because if you look at the literature, the word biology is never mentioned next door to fertility. But I'm teaching it, and I'm teaching students who are probably thinking I'm a bit irrelevant, because fertility is about getting enough nutrients, making sure the soil's okay physically. So I started using the term soil biological fertility. Then you say, well, what is that? What is biological fertility? And I thought, well, that's a bit hard to get your handle on as well, because you've got all these bugs in the soil doing their thing, but what is biological fertility? Okay, so in summary, biological fertility contributes to chemical fertility because it's a nutrient cycling. Chemical fertility can also influence the biological fertility. Biological fertility influences physical fertility. So the hyphae, the organisms, the stickiness of bugs in the soil, the earthworm holes, everything that's going on in the soil can affect the physical fertility. So then we've got, okay, chemical fertility, what do you measure? Well, you measure nutrients, but it's also the nutrients that are cycling with the organisms. So you can't really separate those because it all comes together. And then physical fertility, well, you can measure structure, but part of that structure is the organic matter breaking down, all the gums sticking it together. So you can't really separate the physical and the biological fertility there either. And the whole point is that this is an integrated system. So the biology works with the chemistry and with the physics, and you can't actually separate them if you've got a really healthy soil. And you're heading in the direction to make a healthy soil, but this is where I came from after a lot of thinking. It might look a bit obvious, but if you are teaching in an area of soil where you can't see where you fit, and that's a simple thing like soil fertility. And then I thought about this a long time. So at the end of the day, you can't really measure it easily, and I'll talk a bit more about that as I go. So you've got these multiple functions that are related to microbial processes, and they link to biological fertility, but they develop over time. You can't switch it on like putting a fertiliser on or putting something else on. It takes time. So this is an important point. You can't build soil health by magic. It takes time, and so you have to realise that it's going to take time, so it's not going to be instant. It can be about nutrient replacement, so we've heard that from Colin. You can build that capacity so you put less on, but you still have to replace the nutrient needs for the plant. But it also, a more biological base for supply of nutrients affects the delivery of those nutrients. It's not all front-end loaded, it's happening through the plant life. It might come quickly at the beginning, but there's going to be a continual supply. So biological processes establish gradually, nutrients lost need to be replaced, and there's a more gradual supply. It can still be a flush of supply of nutrients, but it can be more gradual. So this has implications for a lot of things. It has implications for plant physiology. So a plant that's front-end loaded with fertiliser is very different in its physiology to one which drip feeding. It might get a big load at the beginning as well, but the plant is going to react differently. If you have a big overdose of food, you won't feel that good, and if you just eat more regularly, it might be better. It can affect grain production and quality, forage, but the cost. And I think I was pleased to hear a farmer say it can be profitable, because I've said this without the evidence, so thank you, Colin. But I think it is all about the economics. I'm not here to tell people, maybe you can do this, and if your bank balance or your bank manager is telling you you can't do that because you'll go broke. So, but I'm saying that getting this right can be profitable, and I'll go to the experts who can give verification from that. Mind you, in Western Australia at the moment, the Department of Agriculture is looking at this question, because usually the focusing on a more biological approach gets dropped out of an economic model as a whole farm level. I can assure you that's usually what happens, but they're now looking at how that can be included without excluding it at the beginning, without really taking into consideration things like time, building capacity, and resilience is what we're really doing over time. So just bringing you back, diversity of soils, diversity of plants, and there's a lot going on at this level, and we've already heard from Oliver, the pores in the soil for the water to move. But this is an example of a sand, and even a sand can be healthy, but maybe it will be a different measurement of healthiness compared to a heavier clay soil, and I'll come to that in a minute. So if you put it in a picture, the centre of the equation is the roots. And in a grazing situation, you're usually looking at mixed species of plants, so their roots are all getting intertangled. The roots are next to one another. They've got their own microbial community, which might be a bit different, but their hyphae can link in together. And so these roots are happy family in the soil, interacting with one another, and they have organisms in the rhizosphere, which is the area near the root, the rhizosphere community. So near the roots, there's a lot of carbon just leaking into the soil, and that feeds this rhizosphere community. There are pathogens in the soil, things that are going to cause disease. Well, if you've got everything ticking along, you can minimise these guys. They'll still be there, but it won't be out of control because you've got more balance if you're focusing on the organisms. Mycorrhizal fungi are present in all the soils, but more dominant in some than others. So in a canola crop, mycorrhizas go to sleep and they gradually decline over time because they don't have a root system they can colonise. But most plants have mycorrhizas and some develop more than others. But these fungi are really important in nutrient cycling, but also in soil structure. So they have multiple values, but they're quite hard to measure, but they're integral in a farming system. And the rhizobia, if you're growing legumes and you're inoculating, that's well understood. The gold star inoculation system, which is worked out with a lot of critical control, and you can buy it and you know what you're getting, unlike many other things that you might purchase that are microbial. Now, you can't see the details of this, but I'll just tell you, this is a poster made by Gupta from CSIRO. Have you know Gupta? Pardon? Yes, Gupta. And he made this poster. We worked with him on soil fauna many, many years ago. And this is a terrific poster. And if you want a copy, I can get it back. We can put it out to you later. But the point of this is the plant material, roots, kickstart the whole of the soil biology activity. And a lot of people, even microbial people, think bacteria are the centre of the universe, but fungi are also there, and all these animals are in the soil that are very important as a part of this cycling. So you put on the organic matter comes as roots or leaf litter or whatever. The fungi and the bacteria go to town. It's a carbon source. Most of them are dependent on that. Then it just creates a chain reaction of all the other animals in the soil. It's pretty vicious. If you get a microscope, you can see them eating one another, all sorts of things. But that is really very complicated. But they come and go. So you have waves of activity of these organisms in soil, and that's what's happening in the soil. You'll hear more about that later on. Now, way back in those days, with Gupta, we did study in a cropping system, the microarthropods, and of course for most of the year in Western Australia there's none happening, you can't find them, and then as soon as the rain comes, then wow, they're all there to do their thing. So measuring something in soil depends on the time. So if we looked here, we say, oh, there's none. We look out here, we say, oh, there's lots. And that's understanding the life cycles of all the organisms in the soil helps to understand whether you will find something or not. So just finding nothing or a particular organism doesn't mean it's not there. Now, we've done a lot of work on grazing, but in artificial situations, not on the farm. And this is an example of where we've been including the perennials in the farming system. But in this experiment, we had a pair of scissors and we were cutting. So we didn't have any sheep. We were just cutting ryegrass at different rates, different intensities and over time to see what happened to the roots and to see what happened to the mycorrhizal fungi in those roots. So the sugar in the plant is going down to the soil through the roots, but the mycorrhizal fungi in this case are getting that carbon. It's like an electric plug. they plug into the root and help themselves from the carbon. They don't wait till it comes out of the root. So we wanted to know how the mycorrhizas developed on these roots after they'd been grazed. And one of the things that came up was and people know of course, that when you graze plants, you graze their roots. So different species respond differently to grazing plants, but these guys, the ryegrass, graze the tops, you actually decrease the roots a lot. But one of the reasons for that is how much photosynthesis is being sent down there. But in this case, this is a measure of sucrose, so sugars in the root, one form of carbohydrate. So we measured that with no grazing, so that's these guys. There's a lot of sugar in those roots. But with heavy grazing, like here, there's not a lot of sugar. Even though you've got smaller roots, you've also got a lot less sugar going into the soil. So we've already heard from Colin about grazing, and I agree that the grazing really affects the soil community. It affects it by how much carbon's going down, but it also affects by root growth, and as I said earlier, all of this is integrated. So what you do to the top is having an effect. But it's not having the same effect on everything. Different plants respond in different ways. They have a different life pattern. But on the whole, you're influencing with grazing. So that's a really good management tool for building soil communities to do their thing. Even if you don't know exactly every little detail of all those organisms, you know that you can manage them over time. Now, we've done work with soil amendments in dairy. This is a long way from a dairy country, but I've left it in papers. It was a mistake. But we worked on farms. These were big plots. They were put in by the Southwest Catchment Council at the time. The dairy farmers have got a lot of effluent. They want to get rid of manures, whatever. So we measured used DNA to measure microbial communities. Now, this is technology gone crazy. We can now measure all sorts of things to do with microbial communities because the bulk of organisms in soil can't be grown. You can't take them into the lab and grow them up. So with the DNA technologies, you can then get a handle on the diversity, I'll explain in a minute, of what's going on in soil. So in this particular study, one of the things we measured is a pattern of the enzyme capability of these organisms as a community. Now, that's sort of complicated. So we measured the bacterial diversity. So we measured the DNA patterns, basically, of the whole community. And then we went and looked, and I didn't do this, by the way. Bede Micken did this work. So he goes in and he can look at the pattern of the DNA and pick out which pattern belongs to which enzyme. So the DNA is a blueprint for proteins to be made. So out pops all these patterns for enzyme DNA patterns, right? So he can measure this. And then you notice I've got here the sample in winter. We also did it in summer. And then for he got the DNA samples for different enzymes on the DNA of this whole community. And some of these enzymes could split the carbon molecules in starch, hemicellulose, cellulose. So you need different enzymes to bust up these molecules, these carbon molecules. And this is just the amounts of them. And the colour patterns here where we had treatments in this hectare plot. So we had manure added, compost added at a different lower rate, and fertiliser only. But just look at this experiment. Fertiliser was added in all the treatments. So the fertiliser was kept the same and the amendments were added. Now, we wouldn't design an experiment like that because we'd think, well, can we substitute the fertiliser with manure or whatever? But these people were a bit worried about leaving off the fertiliser. What would happen if you left off the fertiliser? So in this case, the fertiliser was added across the line. So for this study, these treatments created some changes in the amounts or the capacity of this enzyme pattern to be reflected in the microbial communities. So it's just, in summary, you've got bacterial DNA as a whole community, and within that DNA pattern you've got enzymes which can break down carbon, and then you've got these treatments. When you manage the soil, you can alter the abundance of some of these enzymes. Now, what that means is another question. I'm not saying what it means, but you're changing the community. So when you add something to the soil, if it's a fertiliser, if it's lime, or if it's something, you can fiddle with the microbial community. And this is one of the ways you can measure what might be going on. Now, another way, yes. Oh, yeah. So the more common way that this has been done is different soils, bacterial diversity measured by just measuring the phyla, the groups of bacteria in this case. Now, this tells you that this soil is dominated by these two groups of bacteria. And when you add these this time it's a different treatment. We've got nothing, and then biochar. Then we've got this mixture of lime, biosolids, and clay mixed in. So you've got different combinations of materials added, and that's why the different colours. But in this case, if you have a look across all these three different soils, and they're sandy soils, by the way, these two groups of organisms are dominant in all of the soils. And there's slight effects of the treatments, so you're fiddling with the community. But within each of these little bars, there are dozens of different organisms. And some of them might be only present in tiny amounts, but they might be very important in their function. So while this approach gives us a lot of data and pretty patterns, it doesn't really tell you it's not a good indication of soil health. It tells you you've got a lot of diversity there. It tells you that if you change your management, you might fiddle them, alter them. But we can't use this and give it to you and then you can decide to do something. So we're understanding what's going on, but this is more of an academic. I mean, I'm an academic, I'm saying this. It's not a very practical thing for you to do, except if you follow your soil over time, you might get some clues about what's going on. So when you look at these data across a lot of soils, it's not that useful, but when you look at it just for your soil or your paddock, you might find something that's trending. It might give you a clue of what's happening. So you can spend a lot of money on analysis. How helpful it is a different question. Now, just a couple of follow-up from this DNA analysis. Some of these organisms, lost a letter there, but some of these bacteria are affected by plants, and in this case, nitrogen. So here we've got nitrogen added, nothing, low level, high level. We've looked at the bacteria associated with the roots of clover and ryegrass, and if you look at this big purple patch, this group of organisms was not really affected by the plant or by the nitrogen in a rough sense, not much. But another group in the same soil, and this time if you concentrate on this bit, you know, this group sitting in the same soil with the other one, there was a bigger effect here of the plant and of the nitrogen. So when you think about this, you say, you know, these treatments are having effects on some groups, a different effect on other groups. So it's such a complex system. When you're looking at this level, this information is interesting. It helps to understand what's happening because you can't grow these organisms. But it's not a useful little piece of information for you to then, I'm going to manage it this way. You can't jump from this information. Now, I already told you that I was a mycorrhiza freak. I've really enjoyed understanding roots. So this is what a mycorrhiza looks like, a bit of root. And maybe you can see some little strands of fungus attached, but mostly you won't. If you dig a plant up, it'll be a mycorrhiza. The fungi will be there, but you can't see them. So you have to cook them up, stain the fungi, and then you can see what's inside. That's what fascinates me. These fungi cannot be grown by themselves without a plant. They are completely tuned in to the plants, but they can grow in most plants. So you don't it's not like rhizobium where you have to get the right bug for the right plant. You have to pick the rhizobium, you buy it in a packet, it's got a label. You can put this on whatever plant legume you've got. But these guys, happy to go into any plant. But more interestingly, most plants let them in. They don't say, go away. And in contrast to a pathogen, most plants exclude most fungal pathogens or bacterial pathogens. They only accidentally let in some that are going to cause them disease. So these fungi are very unusual, and there are helper fungi in the soil. They're a little bit complicated to study. In this case, we were adding soil amendments. So I'm trying to change the soil conditions to see how that affected the fungi. We added a mineral fertiliser, like ground rocky type fertiliser, an inoculant, nothing, and a chemical fertiliser. We put these different treatments in a pot experiment. This is a student who did this with us. And we looked at these groups of fungi, in this case with a DNA measure. These are whopping great big spores. These spores you can see. They're the biggest of all fungal spores. And to me, they're exciting when you see them under the microscope. They've got thick walls. They stay in the soil when it's dry, and then they grow later. So again, the colour scheme for these different treatments. And we were wondering whether some fungi went in some plants more than in other plants. And we wondered whether if you amended the soil, put fertiliser or an inoculant, that might affect how much of these fungi went in the soil. So again, the main groups of fungi, clover, ryegrass. Let's focus on the red line. Anyone colourblind? Yep. So let's start with the bottom one, okay? We're following this one. So this one is a chemical fertiliser, and it was added to subterranean clover and ryegrass to the soil, right? So we looked at these fungi in the roots of the clover and the ryegrass. So we're just looking at this one. So this fungus went into both of them. And actually, with a chemical fertiliser, there was more as a percentage in the ryegrass than the clover. That's a whole other story, but this didn't decrease the fungus in that case. So the chemical fertiliser for this fungus, mycorrhizal fungus, the same fungus went into both. Now, when you go down here, a little bit of it in the clover, couldn't find it here. Go down here, a little bit here, but couldn't find it in that. These are subtle differences in relative abundance, okay? So when you look at that, you find that some fungi will be more dominant in some plants than others. But I say that this subtle difference probably has no effect on their function. So while you get communities of these fungi inside roots, it may not affect their function. That's a long, complicated story, but if there's any questions later, we can talk about it. So it's soil biology test. So are you convinced you can send off your soil and get a soil test and you'll know what to do with your soil? I'm waiting for you to say no. I have a pet thing here about soil test. Measuring something in soil doesn't mean it's an indicator. It's just a measure. You measure pH, it's not indicating anything unless you understand what it means. So here we did it. This is on the web. We can give you the link to this. This is a little booklet we did for farmers in Western Australia about measuring things on your own farm, looking microscopes at what you've got. I think we worked with the Wheatbelt NRM on this, and it was done for you. It's available everywhere. But we were quite interested in soil biology tests, and there's a bit of an explanation in that. Now, am I used up all my time now? Yes? Is that a yes? Yes, it is. So I'm going to stop here, but the last thing I want to say is that we developed a soil health app with the land care funding, the National Land Care Program, and it's free, and it's on your phone, and it's got, if you want to put it on your phone, Android or the other iPhone. And there's a fair bit of downloading. There's some movies, there's various things on it. There's a 100-page PowerPoint presentation you can scroll through. It's information, and then there's a pile of podcasts of me rabbiting on about soil biology, which you may choose to not listen to. But we can replace the podcast if you think if you'd like something put on there. If you want to do an interview with somebody, we can pop up other podcasts. But the videos and the PowerPoint, it's just information that's going to be static, but it's not out of date. This stuff really doesn't get out of date.
Thank you very much.
Okay, thank you.